going to let Simon give the stage to him. Thank you so much, Simon, for taking the time, and we're really looking forward to this.
Thank you very much for the nice introduction, Francisco. I'm going to share the screen with you guys. Okay. Can you see it?
Yeah, we definitely see.
Okay. All right. So thank you everyone for tuning in. My name is Simon Ladouce, and first and foremost, I would like to thank Francisco and his team for the invitation and for giving me the opportunity to share my work with you today. So I will present a study that is demonstrating how cognitive events can be captured in the real world using mobile EEG and eye tracking. But first of all, I would like to introduce the context in which this study takes place. In 2009, Scott McKeag and his colleagues presented their vision of capturing brain and body dynamics when subjects were freely moving by capitalizing on the latest technical developments. So it's important to know that at the time, most of the research methods used in cognitive neuroscience were very cumbersome and did not allow the participants to move much. So the emergence of the portable hardware solutions opened a whole new world of possibilities, and the approach was coined as mobile brain and body imaging, or MOBI in short. So as a PhD student with a cognitive science background, I was very excited by the possibilities offered by MOBI to study human cognition. Not only these methods would allow the participants to move, but, and I think it was even more important to me, they allowed to record the brain activity outside of the lab, directly in the real world. So I jumped into the hype wagon, as I was, and I still am, convinced that the MOBI approach has the potential to introduce a paradigm shift in how cognitive neuroscience research is done by providing new ways to study how humans think and act directly in real life context. So to me, as I began my PhD journey, the MOBI approach appeared as a perfect tool to address long-standing issues related to the lack of ecological validity that was plaguing cognitive neuroscience research of human cognition. So by embodied, I mean how the central nervous system is within a body that is himself within an environment, and the interplay between all these entities. But there was a lot of work to be done, and the initial MOBI studies focused mostly on addressing the most pressing challenges of acquiring brain data during whole body movements. And as such, Gwyn and colleagues proposed novel signal processing methods based on independent component analysis to disentangle brain signals from gait-related noise. For this, they suspended a traditional EEG system, very high density, by the way, as you can see on the figures. And this system was placed on top of an individual that was standing on a treadmill. They presented visual stimuli on a screen, on a monitor in front of the participants, while the participants were either standing or walking at different paces. What they did is that they demonstrated that neural responses related to visual, to the processing of visual stimuli could be effectively recorded when participants were walking, and that the quality of the data after the application of their ICA-based artifact removal methods was much improved, as you can see on the bottom plots. So using a similar setup and signal processing approach, Grumman and his colleagues demonstrated that transient changes, so features in the EEG data related to the presentation of visual information, could be as well extracted when participants were walking on a treadmill, either slowly or briskly. So there is a common point between these early MOBI studies, and it is that they use the same time domain feature of the EEG signal to benchmark the data quality. They all use the P300 even-related potential. And as suggested by its name, the P300 is a positive deflection observed in the EEG following around 300 milliseconds the presentation of task-related stimuli. And the Ph.D. has been shown to be most prominent at parietal sites, as you can see on the scalp map. Can you see my cursor, by the way? Maybe.
Your what? Your the sounds?
No, here. Can you see when
the cursor? Yes,
yes,
we can.
Yes,
definitely.
yes. All right. Okay. It will be easier for later parts of the presentation. So yes, it's mostly prominent at parietal sites, and it has been linked with attentional and memory processing. The amplitude and the latency of the P300 are commonly interpreted as reflecting how much cognitive resources are selectively allocated to the processing of information. So this signal was a perfect candidate to validate mobile EEG data, as it is quite robust and can be reliably elicited through auditory and visual audible paradigms. And those paradigms consist in the presentation of stimuli that need to be attended by the participants, which are presented in the midst of non-target stimuli or task-irrelevant stimuli that are just distractors. So following these early studies, Devenos and his colleagues decided to go one step further in the direction of the MOBI vision by taking a portable EEG, this time for a walk. So they demonstrated not only the feasibility of recording auditory P300 during locomotion, but also highlighted a difference in the amplitude of the effect between conditions in which the participants were either sitting still inside or working outside. So you can see that these are the ERP waveforms elicited by the target stimuli, and in blue it is the P300 response to target stimuli when people were sitting inside, and in red it's when they were walking outside. In a follow-up study, they replicated the results, but this time the authors also assessed whether this difference between the two conditions could be explained by a difference in terms of noise levels. But the lack of significant difference between the sitting and walking condition in terms of noise level suggested that the reduction of P300 amplitude was not merely the result of a lower signal-to-noise ratio, but was rather reflecting maybe the additional cost in cognitive resources of being in motion. So that's why later studies have investigated cognitive motor interference and its effect on P300 amplitude. Zink and colleagues and Scanlon and colleagues had very interesting paradigms where they asked the participants to cycle, either cycling inside or going outside. And what they showed is that in comparison to being still and not cycling at all, there was no reduction in P300 amplitude when they were cycling inside, not moving through space, but there was actually a substantial difference, a substantial reduction of P300 amplitude when they were moving around outside. So these findings suggested that the attenuation previously reported was not so much linked with the physical act of cycling or walking, but was rather related to the higher cognitive demands of being outdoor in an ever-changing and dynamic environment. So this is quite interesting, but there were still a lot of unknown variables, and the factors underlying these changes were still very much unknown. So what was exactly explaining the P300 attenuation observed during real-world behavior? Is it something that is purely linked with the external world, or can it be explained in terms of motor demands to act in the real world? So that's the reason why we wanted to carry our first experiments on the topic, and in order to gain a better understanding of the factors underlying the modulation of the P300 amplitude, we carried a series of experiments. That's the incremental experiments that Francisco was talking about. So the first experiment was aimed at replicating the previous findings in a more systematic manner and characterize the difference between standing and walking conditions. So in the standing condition, the participants were simply attending auditory stimuli and were instructed to silently count the number of target stimuli that were presented. They performed the same task while walking, but the walking condition was different in several ways. The participants were engaged in the physical activity of walking, of course, but incidentally, they were also moving through space, which in consequence, they were experiencing a much larger quantity of visual information. So as for the results, there was no surprise, no replication crisis in the P300 MOBI literature. We found the exact same attenuation reported in all the previous studies in which participants were walking or cycling, which motivated us to explore further what was causing this difference in a following experiment. So this was suggesting that there was a reallocation of cognitive resources happening. So we investigated the contribution of walking and moving through space on the reduction of the P300 amplitude using a factorial design, along with the initial standing and walking conditions that were included in the previous experiment. We also had a treadmill condition in which participants were walking but not moving through space to isolate the impact of walking activity on the P300 amplitude. And we also had a wheelchair condition during which the participants were wheeled down the corridors and were experiencing movement but not walking themselves. So in order to isolate the impact of motion on P300 amplitude. The results were quite interesting. It appeared that it was not so much the walking activity that was explaining the reduction in P300 amplitude, but rather the displacement through space. An interpretation of these results could be that walking is a fairly undemanding task for healthy subjects. We do it quite implicitly without having to engage much cognitive resources. But it remained unclear what about moving through space was grasping so much cognitive resources away from the auditory audible task. Was it the increased flow of visual information, or the increased amount of inertial input that needed to be processed by the vestibular system during the motion that was explaining this this reduction of P300. So to address this question, we devised a third experiment. We had a control condition here in which the participants were seated, looking at a gray wall for 6 minutes while performing the auditory audible task. Quite a stimulating experience for the participants. And this condition was in contrast of the previous wheelchair condition in which the participants had both high visual and vestibular input. And we used, again, in a factorial design, intermediate conditions of motion and visual input. So a pseudo-blindfold condition where participants were wheeled down the corridors, experiencing full vestibular input, while looking at a video recording of the gray wall. So nothing really interesting visually for the 6 minutes. And another condition where they were not moving, they were just sitting still. But this time they had a video recording that was displayed on the VR goggles of someone, first-person video recording of someone being wheeled down the corridors. So experiencing a complete experience of visual flow. The results were this time again quite interesting in the sense that both aspects, so the motion, the vestibular input, and the visual input seem to contribute to the P300 amplitude attenuation, but it seemed that it was mostly the visual input that was reducing P300 amplitude. So all these findings are hinting at the interplay between bodily experiences and the dynamic integration of multimodal information. And they also bring empirical support to the embodiment theoretical framework, which is another motivation to strive toward higher degrees of ecological validity. And I think it's the take-home message of those 3 experiments is that they showed how pervasive the visual modality is for human locomotion. So that's nice and all, but you know, the incremental abstraction across the 3 experiments that I presented was not exactly in line with the way MOBI pioneers maybe had, nor did I envision the study of human cognition in the real world. So this was the theory and the point where we wanted to strive toward to, and we ended up with experimental conditions looking like this. So as a progression towards more greater ecological validity, I wanted to get back to the study of human cognition truly in the real world. So I was asking myself the question, can we capture such neural markers of visual processing, such as the P300, in the real world? And this question was quite topical because to date, MOBI studies have mainly resorted to the presentation of artificial stimuli through computerized paradigm. That was the case of my experiments. We were presenting tones that were quite artificial through a computer, while participants were performing were walking or being wheeled down the corridors. So most MOBI studies have either used computerized paradigms, interactions with experimental apparatus taking place within highly controlled environment, as was illustrated by the initial studies that were having participants walking on a treadmill, and experimental protocols that were characterized by the repetition of prototypical behaviors. So an important step toward the realization of the MOBI vision was and is still to transition from computerized paradigms that dictate participants' percepts and responses towards the recording of more naturalistic and spontaneous behaviors. So the question, therefore, is, can we make this leap toward greater ecological validity and study cognitive processes in response to natural experiences, natural cognitive experiences of objects that are integral parts, that are embedded in the real world. And actually, I found out that some other people had thought about it already, and that the answer was yes. So Vicente Soto and Anna Roberts in Liverpool explored the feasibility of recording neural signatures of economic value and emotional phase processing in relation to stimuli displayed in the real world by presenting either faces or objects on boards that were placed around corridors. So they had the participants to move from one board to the other, to look at a fixation cross and to make decisions based on a certain instructions. And they used mobile eye tracking to retrieve the timing information about the onset of visual events and timestamp the EEG. These studies were actually like doing the step toward greater ecological validity that I had in mind. Although it can be argued that the stimuli used in these studies are still a bit artificial. They are not naturally part of the environment. They are pictures on the walls, and they are not. And also, the participants had to maintain their gaze for a certain time when they selected a specific stimulus, specific visual information, in order to make the analysis of later EEG data not contaminated by eye movements artifacts, which is also introducing some bias. And lastly, they had to always go back to the fixation cross between trials. But nonetheless, they provided first evidence that N170 ERP component related to phase processing could be acquired in the real world, and also the P200 ERP component related to attentional allocation and semantic processing could be also recorded in naturalistic or almost naturalistic settings. So I basically refined, after reading those papers, my question. And this time I was asking myself, can we capture these neural markers of visual processing, such as the P300, while participants are engaged in everyday life behaviors and maybe not doing a very specific task that might be a bit arbitrary or completely artificial, such as looking at boards and reproducing the codes of laboratory experiments. While I was brainstorming about ideas of real-world experiments that would combine all these aspects, I was inspired by some books that I had when I was a kid. So it's the Where is Waldo books, in which you have to find that guy over there in crowded scenes. And I've actually found out that there is a whole literature that is doing visual scene search task, which is quite similar to what you would do when you try to locate Waldo in one of these pages. So they use eye tracking information to have an idea of where people are looking at and what kind of information is being processed. And then they can derive the timing information of cognitive events based on fixation on specific objects and on the target object. So to me, it seemed that it would be maybe sensible to think of a visual scene search task as a good analogy for a P300 oddball experiment, in the sense that when we finally locate a Waldo, the task-related information, we should expect a specific selective attentional processing of that information, which would be reflected as a P300. So I initially thought about taking my participants to the supermarket and go through a grocery shopping list. They would have to basically find goods in the alleys. But it was quite impractical, as the University of Stirling is a bit remote, and we preferred to spare our participants the self-conscious experience of wearing a swimming-like cap and funny glasses in a store full of people. But fortunately, the university was equipped with a glorious library, which provided the perfect environment for a challenging visual search task that every student had to go through at some point in their studies, finding a book. So let's do some book hunting. But quickly I realized that designing such an experiment comes with daunting challenges. A first important challenge is the loss of control over the experimental environment when recording in the real world. And you know, anything can happen anytime in the real world. That's the beauty of it, but that's also the problem of it. So the quality of the signal can be is much more variable across the recording. You know, a book that the participants would be looking for might have disappeared as it was taken by another student. Other things like light exposure and temperature condition may vary throughout the day, and also across space in the library. So many things would differ. The library may be more or less quiet as it gets busy with students and so on and so on. So we needed to make sure that the cognitive function or specific processes that we want to investigate are effectively captured, and that the measures are valid and precise. And I think that to address this issue regarding the loss of experimental control and the higher degree of freedom during naturalistic behavior in real-world environment, Paradigm and his colleagues have elegantly proposed that we should progressively transition from computerized paradigms to the study of cognitive processes in relation to embodied experience within the frame of scalable experimental designs, which means that such studies would include several experiments ranging from highly structured to maybe semi-structured or completely unstructured experimental design, which would work together as complementary approaches to study cognitive phenomena, providing both the reductionist abstraction that is needed to isolate cognitive processes with precision. So that's what we did in our third experiment. We had very artificial but precise experiment, and but also putting the findings of such well-structured experiments to the test of more naturalistic behaviors and context. So this approach is further illustrated by a recent paper from King and Paradigm through its application to the study of neural and cognitive underpinnings of art making and observation. So inspired by the scalable approach, we opted to design a study contrasting two experimental conditions, a structured condition and not completely unstructured, but much less structured condition. So the first condition consisted in a classic visual oddball paradigm, abstract visual stimuli, blue squares, and red circles, I think, yeah, red circles were presented on a screen and on a tablet, and the subjects were instructed to count the number of occurrence of red circles. So in contrast, the second unstructured or semi-structured condition was maximizing ecological validity by getting as close as possible to the recording of a natural behavior taking place in the real world. So we asked our participants to perform a scripted but realistic behavior, which was finding books in the library. So through this leap across the ecological validity continuum, differences related to embodied experiences could be explored, while the conceptual and methodological gaps could be uncovered and hopefully dealt with. So I will give you an example of what the tablet condition looks like. You can see that the stimuli are quite artificial. They were presented as a series of discrete stimuli. You can see the small dot over there is actually their gaze that was recorded with eye tracking glasses. And of course, we were recording the brain activity using EEG. And the maybe more interesting naturalistic visual search task is the following. So the following sequence shows one trial in the library condition. So first, the participants grab a paper with the instructions regarding the location of the book in the library, its title, and the position on the bookshelf. Then the participants walk to the location. When the participant arrives in front of the shelf, they start scanning. When they find the book, they just return to the starting position to get instructions about the next book to find. So the participants had to repeat this 40 times in order for us to have sufficient data for ERP analysis that requires averaging procedures. And we collected the data on 24 participants. So a second challenge that comes with recording brain data during naturalistic behavior is how to define the onset of cognitive events for the library condition. So in the absence of external and artificial experimental probes, we didn't really have a very accurate idea of the timing of cognitive events. So in contrast to the tablet condition, in which the timing of the events can be derived directly from event markers with very, very high temporal accuracy, the definition of cognitive events poses several conceptual and technical questions for the library condition. So when do cognitive processes happen in the real world, and how can we timestamp and segment the data accordingly? Two very important and non-trivial issues that need to be addressed in order to actually extract meaningful information from the EEG data. So this is where Waldo comes to the rescue, just as people did in the visual search literature. We can use eye movements as contextual reference points to segment and analyze the EEG data. And within the frame of the study, the timing information of the initial fixations on experimental objects, which are the target book covers, can be used to precisely delineate time periods of interest in the EEG data. So here is an example of raw eye tracking data superposed to scene recording during the library condition. So as you can see, it's pretty quick. And the idea is to manually review the data. We can retrieve the timing information of when a book was visually explored, and basically timestamp the EEG data at that point. So consistent with the fixation-related potential approach used in visual search task, we chose the first fixation on the target book cover as that timestamp. In order to investigate the P300 effect, we also needed to look at brain responses to non-target objects and to be consistent with the tablet condition, which used a 1:4 ratio for target stimulus presentation rate. We extracted the timing information of 4 fixation on distinct books preceding the initial fixation on the target book. So basically the gaze pattern would be like this, and at those points there would be fixation on distinct books. So we would use these timestamps as a non-target stimuli to have a way of comparing target and non-target stimuli in the library condition as well. So you can see that all the EEG data is basically time-stamped based on the computerized paradigm. Here you can see it's very consistent, and here you have the timing of all these events with the variability in terms of fixation duration length, fixation duration, saccade, and so on. So the study of cognitive phenomena in the real world prevent us from using artificial probes, but there is also we can fix that by using the eye tracking data to retrieve the timing information of cognitive events. But the last challenge was how to deal with the noise in the data when people were walking and moving around. And as mentioned earlier, the development of signal processing methods to deal with artifactual sources of noise in the EEG data is still a very much ongoing work, but there are, however, several tools that have shown to be effective in dealing with ocular artifacts, and so on. So on shores of giants, we implemented state-of-the-art methods to our processing pipeline to minimize the contamination of EEG data by non-brain sources. And so we were quite confident in the design of our scalable experiment, our ability to extract visual events based on eye movements. And we had a fancy processing pipeline. So we started collecting data and were hopeful to capture neural signatures of visual processing in response to the detection of objects that were actually embedded in the real world. So did it work? Not really. So as you can see in the top left corner, the target stimuli of the tablet condition here successfully elicited the typical P300 waveform, whereas absolutely nothing was found for the library data. So both the target and non-target stimuli elicited a flat line. This is further illustrated on the right plots presenting the distribution of P300 amplitude for both conditions across part spans. So each point is a part span, and its mean P300 ERP amplitude for the tablet condition. So you can see that the target stimuli had a much higher P300 amplitude than non-targets. But here for the library, it was basically the same, 0 for target and non-targets. And in terms of spectral dynamics, it can be seen from time frequency plots on the bottom left that target stimuli elicited a strong increase of activity in delta and theta bands for the tablet condition, whereas only a marginal increase was spread over time in the library condition, and was not statistically different from non-targets, by the way. So why didn't we find the P300 condition? Could it be because neural markers of visual processes recorded in the real world are completely different from those elicited by computerized paradigm? This could be the case from a conceptual point of view. Experimental protocols evoke phase-locked neural responses by presenting series of discrete stimuli. And a pending conceptual question is whether these artificially induced neural responses to discrete events are also present during natural behaviors, and it could be the case that they don't. So the definition of a cognitive event in the real world is much less clear-cut than in computerized paradigm, and objects in the real world are permanent in the sense that they are always there. So they are not appearing out of thin air, and they might not evoke the same phase-locking responses in the brain than the stimuli presented through computerized paradigms. So another possibility was also that the brain signals recorded in the library was more noisy than the tablet condition. But this hypothesis was, however, not supported by data quality metrics that we compared across conditions and that were quite similar. And we applied exactly the same processing pipeline to data from both recording conditions. Lastly, the definition of cognitive events based on initial fixations may either be invalid or inaccurate. So we manually reviewed the scene recording, as I said, which has a sampling rate of 25 frames per second. That means that even if the initial fixation on the experimental object was corresponding to the actual onset of cognitive processes, there would be a variance of 80 milliseconds. And considering the millisecond scales of ERP components, averaging at trials whose latency has such a high variance may completely eliminate the effect by canceling out the single trial responses. So in addition to this imprecision, the validity of using the first fixation on target can be questioned, and maybe other a priori methods could be used as the onset of preceding saccade or the previous fixation. On top of that, recent studies on reading and visual search paradigms have demonstrated that semantic information was already processed when the visual stimuli entered parafoveal or even were present in visual periphery. So we could use also other approaches to retrieve the actual onset more precisely of such cognitive events. So we were a little bit curious about what was explaining this lack of P300 in the library data. And when looking at the EEG data at a single trial level, we observed that in contrast to the highly time and phase locked pattern of EEG signals in the tablet condition, as you can see here, the library data exhibited responses that shared some properties of the P300, increase of activity, delta increase, but were widely spread in the time domain. And as a consequence, not phase locked or time locked, which didn't result in an ERP response. So we wanted to assess whether or not the P300 was actually there somewhere, but maybe time shifted. So we used a method called dynamic time warping to assess the similarity between the tablet P300 ERP response and the library P300 ERP response, despite temporal lags. So it's basically shifting a template that will be based on tablet P300 ERP for each individual subject. So we compute the mean P300 ERP for one subject. We select the channel at which there is a maximal amplitude. We extract P300 time frequency features, and we use those features to maximize a template model that will be slided along the library data. We did that for both target and non-target epochs, and what we saw was that significantly higher similarity was found between time warp target signals than between template and non-targets. So that was suggesting that there was P300-like ERPs present in target library data, but not in non-target library data, and that they were shifted in time. So we needed another method to realign library ERP data, and this one was a cross correlation. So basically we computed the same template of P300 tablet data for each individual participant. And we computed this sliding dot product to have a measure of similarity between between the template and the library data. And we found, looking at the probability density function, we could find the point at which there was the best alignment between the template and the signal, and retrieve the lag between the template and the original library data to shift the timing onset, the onset timing of cognitive events. So this plot is showing the distribution of temporal shift, maximizing the cross-correlation coefficient across all trials of all participants. And the different colors represent the type of gaze dynamics they relate to. And when we go back to the eye tracking data and extract the type of eye movements that were performed at that moment. So these large distributions actually preceding and following fixations and saccades further highlight the limitation of using a priori approaches, such as fixation of onset, to retrieve cognitive event timing when people are doing a naturalistic task. So we lag corrected the library data. So we computed the ERP again, and ta-da! Here you can see, we can find back our tablet ERP, and this is the lag corrected library data that now has typical latency, but also a prototypical spatial distribution, and has the same spectral features as an ERP response that is elicited through computerized paradigms. We didn't find any differences in terms of the amplitude of the P300 after the realignment of the data. So in conclusion, this study is showing the feasibility of recording neural markers of visual processes elicited by the detection of permanent objects embedded in the real world. It stresses the importance of multimodal recordings and how eye tracking and EEG can help each other to study human cognition. And also it highlights the relevance of scalable experimental designs. And that's it.
What a trip! Thank you. Yeah, that was that was super cool. Ending. I was a little bit afraid because I was like, when we first talked about this thing, you present some of this data in Serbia, the Serbia conference.
Yes.
And that ended with that kind of like bittersweet ending. It was like kind of like, yeah, we have the data, seems cool, but like there's nothing there. I'm so glad to see that you actually found out that way to actually recover that ERP. That's amazing. Have you tried doing any of the of these regression models for the unfold toolbox? What they suggest on that? Have you tried that, or you didn't try that one?
Yes, actually, there were two tools at the time after visiting Serbia and meeting with the researchers. They gave me some good feedback and some interesting suggestions using Benedict Anger toolbox. So unfold. I've applied it to the data, but actually the variance in terms of the latency shift was much larger than what the models could account for. So I've also tried the ride modeling toolbox, but this toolbox was working best if you had a rough estimate of the starting or the ending of the events, and of course it wasn't the case. So these approaches were quite limited to account for the wide variance in terms of latency that we were dealing with in that experiment, I'm afraid.
Yeah, but the solution was quite cool, and I guess that is data driven in a way. So if you have good profiles of what you think you should have, you can actually.
Well, actually, I found out about the cross correlation quite a while ago, but I was very skeptical about using it, because maybe you just perform a template and then you slide it across any kind of data, and then you have something that looks like it, but that doesn't allow you to call it that kind of signal. So that's why we had this two-step kind of approach using dynamic time warping to look at the similarity between our template and neighboring signals. We didn't slide it across the whole recording. We used our a priori fixation related epoch. We epoched slightly larger around that timing, and we slided the templates, and we computed those measures.
Sounds amazing. I'm going to leave the microphone open or the chat open for anybody that wants to ask questions, and we can continue talking later. So are there any questions from the audience or some other people that wanted to ask? Great. More questions for me. Daniel, do you have? I can see.
Yeah, I have some questions. I don't know if we are like talking right now or yeah. Okay. So this is very interesting. It's more very nice work. I imagine also it's a lot of work, and I can imagine facing that flat line and trying to analyze data, learning new ways of doing it, and so it's a very amazing work. Yeah, I actually didn't quite completely understand when the ERP, the library ERP appears at the end, right? So you have this template that you get from the tablet ERP,
Yes.
And are those the same subjects?
Yes, exactly. And I think that's actually illustrating the relevance of scalable experiments, because you can actually use the template that you have acquired from a much more controlled paradigm, and then you can see, you can compare those signals, those templates to the data that you have acquired in more naturalistic settings. And that was very much the inspiration of that analysis approach as well.
Yeah, and
it was specific to the subject, of course, yes.
Yeah, but how exactly do you sort of control that you're not like you're not putting what you want to end up with, and you're not entering into your signal? That's the part I didn't really get. Did you try, for instance, using the template from one subject to using it at a different subject or stuff like this? Or
yes, that's something that is commonly done in BCI actually, where we record a lot of training data, and sometimes we've got better results by averaging the training data that is collected over a lot of people. But actually, in that case, I've tried it and it wasn't working so well. I've actually, there was differences in terms of the channel that was most responsive to the P300 across subjects, and also in terms of the latency the waveform had slightly different shapes. So by using a more general model that was including more participants, it wasn't improving the similarity measures.
So this subject A uses template from subject A.
Yes.
And the idea is to use it in a slightly longer window.
The window was larger because I was suspecting that there was quite a lot of variance in that, because conceptually we were not quite sure that that the cognitive events were actually starting on the initial fixation. It could have happened much earlier, because the processing of that information could have happened already when the book cover entered the peripheral vision. But it could also be the case that, you know, they were scanning, they were going from one book to the other, and sometimes they might just skip the target book and just go back with a very ballistic eye movement to this one. So sometimes it might take a little bit longer. And I wasn't surprised when I used the lag correction timing, and I went back to the eye tracking data, because I was curious to see if there was some consistency in terms of what was happening in terms of eye movements at that time. But actually, sometimes it was during the saccade, sometimes it was during a fixation, preceding or following the initial fixation on the object, but it was widely spread with normal distributions. So yeah, I think that it was just the limitation of our naive a priori approach, and that fixation related potential for with an equipment that has such a low frequency resolution, sampling rate, I mean, is quite limited for many reasons. Also, it's not like the Where is Waldo, where everything is static. These are dynamic scenes. There are whole body movement, there are head movements on top of that, and your eyes are moving into your head. So I mean, there is a lot of degrees of freedom to account for.
Yes. So then your sort of interpretation of the story is that there is a component that is similar to the one that you see in the tablet, but sort of the onset of that component when it enters like the visual processing stream or something of the sort, it would depend on whatever the subject is paying attention to or whatever. So it's something like this. But so you have to find it at different moments for each subject.
I don't know if it's if this variation is really in terms of subjects, or is more something across trials even. Another, you know, example of the higher degree of freedom is that, you know, I was asking them to go to a specific location to look for a certain book, but the neighboring books could have very different visual features from one trial to the other. And as has been seen in the visual search literature, if other distracting elements in the visual scene are very colorful or very bright, they might attract attention. They might have an impact on later processing. They might act as a cue. They might actually distract or grasp away cognitive resources. So I think that it's quite difficult to have only one definitive answer about that.
Yeah. Yeah, in your paralleling, how did the subjects learn about the target? Like they were told like the name of the book, or they were shown?
Yes, in the video that I presented, they first pick up a sheet of paper on which there is the alley, so the location in the library where they have to go, and then the title of the book that they are looking for, and then the bookshelf. So the second bookshelf from the top and the third bookshelf. So it was always bookshelves at eye level. So we didn't introduce even more neck muscle artifacts. I mean, yeah, they had the instructions on
Oh,
paper
okay.
that
But they were not
carrying with them.
So they were not really looking for a sort of a visual image. They were looking for a name or something.
Yes, yes, and that's
okay.
that's something also different. They had to read. So in terms of the latency, it might also have had an impact.
Yeah. Okay.
There's a question in the chat. It says,
Okay.
in the natural environment, is it possible that the subject can perform a motor task, for example, touching the object, when identifying the target, in order to bring the biological signal closer to the identification of this object? Or would it be something unnecessary? Or does it make the signal dirtier? Mm-hmm.
It's a fair point, and actually one of the toolbox that could actually help me correct for the latency variability was based on that. It was based on paradigms that were presenting information to the participants and expecting a response, so a motor response to it, either a button press or so on. And the toolbox was using the onset that might be variable and the offset that was fixed to basically fit ERP components that were expected to be found at those specific timings. So I think that it's a quite it would have been a good way to deal with that problem. And actually, in the video that was just made for the purpose of illustration, I had the participant to touch the book, but they were not doing it in the experiment because they didn't have to do it with the tablet condition. But we could have made both conditions requiring a response, a motor response from the participants, and it would have been helpful, actually, I think, in relocating the timing of cognitive events. But yeah, and about making the signal more dirty, I guess it depends on how much movement is involved for the performance of that task. But I mean, we've been collecting data in a lot of real world behavior context, and it really depends. But we can deal with a lot of artifacts.
Good, thanks. Any other questions from the audience? Okay, so I would like to go back to what Daniel was talking. Did you try to grab like some random piece of data? So for example, same PZ electrode, but now let's just fake a fixation and do the correlation thing, just to like test if you put anything into it and you do the dot product with an ERP, would you get some sort of like an ERP-ish signal? Did you try something like that, like a null model in a way? Yeah,
that was actually the reason why we applied the same procedure to the non-target data. So since we were not expecting the P300 to be found in response to those non-target visual stimuli, we applied it to it, and we didn't have a P300 looking response as a result.
I see. So,
but it was still finding. I mean, it's optimizing the temporal shift to make it look as close as possible to the template. But maybe the thing that is the closest to the template is still very, very different from it. So by averaging all these time shifted signals, we didn't have anything else than a flat line again. So yes.
Yeah, that sounds good. I mean, yeah, I was thinking like basically the non-target fixations were actual fixations. So if the P300, but you're right. If there was, you were not expecting a P300 in those fixations, so there wouldn't be. I mean, if the P300 you found was just basically an artifact of the method, you should be able to find it in the non-target fixation for sure. Yes.
Yes.
Yeah. Okay. Any other questions from the audience? Because otherwise we can just keep it here, and Simon and us, we will talk shop about some other stuff a little bit later. So the public talk, in a way, it's over. if you guys don't have any other questions. So thank you so much for coming and for
Thank
being
you, everyone.
in the talk. Yes, thank you, Simon, for the talk, and we will well see you guys at some point. Thank you so much.